So the process, don't forget, has three parts to it. You've got glycolysis, the Krebs cycle or citric acid cycle, and the electron transport. Glycolysis happens in the cytosol, or that fluid portion of the cytoplasm, and splits the glucose molecule into two pyruvate molecules. We didn't get much energy out of it, certainly not enough to power a cell, but we're preparing that process of stripping the energy from this glucose a little bit at a time. Next, we have the citric acid cycle. And the citric acid cycle's job is mainly to get rid of the carbon from this glucose. So you've got 6 carbon molecules in each glucose, and that's a major waste product. It's got to be gotten rid of. Okay, so the Krebs cycle is going to take that carbon from the glucose, switch that to carbon dioxide so that we can transport it through our blood and breathe it out. Okay, once we have it broken down into carbon dioxide, it's a very easy waste product to get rid of. Okay? So the steps to that process include, first, pyruvate has to enter the mitochondria, where it is immediately switched, okay, through some work, okay, a little coenzyme here. We're going to switch from pyruvate, that is going to be adjusted to acetyl-CoA. Okay, so this does very little for us. It does reduce an NAD+. Okay, however, there's no ATP produced, but it's just the next step in the process. Pyruvate cannot be used directly in the mitochondria, so we switch that to the acetyl-CoA. Acetyl-CoA is the start of the citric acid cycle or Krebs cycle. Now, the cycle is going to, when we have that acetyl-CoA produced, the cycle is going to start and stop with the oxaloacetate. Let's take a look at the cycle. It is all of this broken down by Mr. Krebs, and just like glycolysis, every step in the process is known. What is being broken down or switched into what kind of enzyme does that job, where exactly do we produce, okay, where exactly do we produce the carbon dioxides, where exactly do we reduce the NADs, where are we getting ATP out of this. Okay, so all the steps in the process are known. The number one thing to know about this is it is cyclical. Starts and ends with the oxaloacetate. So once we have the acetyl-CoA brought into the mitochondria, that can jump into this cycle and this process can happen. Notice we're going to get rid of molecules of carbon dioxide in several places. Okay. We are going to reduce not only NAD, but also its cousin molecule, FAD. So if NAD is niacin, the nicotinamide adenine dinucleotide, okay, FAD is one of the flavonoids. It's a B2, vitamin B2. So the FAD now is going to be reduced to FADH2, and both of these will serve as electron carriers for us. We'll show you what those electron carriers do at a later time. Notice here, we're not producing a lot of ATP. Okay, the job of the citric acid cycle isn't to produce a lot of ATP. It's to get rid of the carbon dioxide, okay, get rid of that carbon waste product, and to charge up our NAD and FAD coenzymes. So what do we get from the Krebs cycle, the citric acid cycle? Okay, what we get is, well, what we get here now is we do get one ATP out of the deal, okay, but we get 3 NADs reduced, okay, so 3 NADH. We get 1 FADH reduced to FADH2, and we get every turn of that cycle eliminates a carbon dioxide for us. The Krebs cycle.